Okay, so welcome to CRM. CRM is the Centre for Regenerative Medicine. It's a research centre as part of the University of Edinburgh, where we study multiple aspects of stem cell biology, tissue regeneration, and with the ultimate aim of advancing human health. So this young lady that you can see here in the front of this image is Sophie. She's a PhD student in the centre, and she acts as the tour guide for the virtual tour. But today you're going to have the pleasure of me and my research group narrating your journey. So the people that you'll see throughout this video are not part of my team, but as I said previously, you'll hear live from my three PhD students, Sonia, John and Ella, who are all here right now on this call. And you'll also hear via video from my postdoc, Cecilia. So I'm just going to take you through into our lobby. So this is a communal area where people often meet. So here you can see Robin, who's actually working behind the science today, who's one of our public engagement professionals, and he's talking with Sophie here. So within the centre, we have several hundred scientists and clinicians studying the processes of development, how to harness the potential of stem cells, through to better understanding diseases such as cancer, multiple sclerosis, liver disease, and Parkinson's disease. So we're now going to take you from the lobby upstairs to show you some of what happens in the centre every day. So our building is almost 10 years old now, and it was designed especially to house laboratories and to facilitate a good working environment. So the building was designed as concentric rings with the offices, like you see here, located on the outside of the building to concentrate daylight and natural ventilation on the spaces that would benefit the most, and to also maximise the incredible views that we have out of the building. And then we end up with the labs and the more specialist areas on the inside of the building, which you can kind of see here. And in this image, Sophie's talking to Professor Ian Wilmot, and Professor Sir Ian Wilmot led the team that created Dolly the sheep, who I'm sure you've heard of in terms of stem cells and kind of genetic manipulation, pioneering advancements in science. And Ian was the founding director of the centre in 2008. Ian's since retired from his role as the director, but he still plays a really integral role in the centre. And he's actually the namesake for the annual Best PhD Presentation Prize, which was this year won by one of my research group that you're going to be hearing from later, John McKendrick. So we're now going to walk along the corridor and enter the lab. So I'm just going to switch scenes so we can go into the lab. Okay, so from here on, I'm going to tell you a little bit more about what my lab's doing and how we use this building to facilitate the research that we're doing. So as I hope you've kind of gathered from my talk this morning and from the Q&A session, and also from John being involved in the Q&A session, our research is really focused on developing regenerative strategies for damaged salivary glands. So here you've got a view of the inside of our lab. So this is the lab that we work in. These people are our neighbouring lab mates. We actually work just the other side in this bay here. And this is where we undertake all of our kind of day-to-day experiments that don't require anything that's particularly sophisticated. The labs are set out in this sort of open plan manner that you can see here. And this has adjoining bays that facilitate collaboration and discussion. And then we have dedicated rooms off the main lab for more specialist techniques. So one of the initial methods that we use to understand how the salivary glands change after damage is to prepare small pieces of gland and then look at these small pieces under a microscope. So this process is known as histology. And we're very lucky that the centre has a fully equipped histology lab, which you can see here in this view. And at this point, I'm just going to pass over to Sonia, who's going to tell us a little bit more about how she uses histology in her project.
Hi everyone, my name's Sonia. I'm a first year PhD student who joined the lab in April of this year, coincidentally just before lockdown happened. So my project focuses on whether cells which are forced to become prematurely aged and therefore stop dividing are important for the regeneration of the salivary gland following irradiation damage. So as you can see here, we're in the histology room. And as Elaine said, histology is known kind of as the study of tissues and their structure. And as we know, the structure of tissues is very important and it's related to their function. So for example, the salivary gland is very important in producing saliva, as we all know. However, following radiotherapy, the salivary gland is damaged, and this causes changes in the tissue, which we can look at under the microscope. So the first thing that we do when we receive the salivary gland is that we actually fix the tissue. And this is the process of kind of soaking the tissue in a chemical. And this allows us to maintain the salivary gland and all of the microscopic components in the state they were in when they were removed from the mouth or from the patient. And this really allows us to see a really accurate view of how the tissue was when we removed it. So the next thing we do is we then embed it in one of two things, either a wax or a compound called OCT, which is then allows the tissue to become frozen. And once we leave these substances to harden, we end up with two blocks. So we have either a block of wax with the salivary gland in the middle, or we have a block of frozen, just kind of like a frozen block with, again, the salivary gland in the middle. And then what we do is we section the salivary gland into very thin slices. And we do this either using the Microtome, which is a machine which allows us to cut thin sections of wax, which I think Elaine is going to kind of show you here, or we use the cryostat, which is used to section the frozen tissue. And this machine is really good for keeping everything at a very cold temperature. Then what we do once we've got the slices is we transfer them onto microscope slides, and then we can use this to stain for different features of interest. Another thing that we do in the lab is we actually embed the gland in a gel. And this is something that myself and Chechi, one of the postdocs in the lab that you would have heard Elaine talking about earlier, have actually been working on in the last few weeks. And this machine that we use for sectioning the gland embedded in gel is called the Vibratome. And this is actually found in the main section of the lab. And this works in a very, very similar way. We section the gland in a gel, and we use a vibrating blade inside kind of like a chamber of kind of water with antibiotics to prevent it getting infected. And what's really good about this is that we can actually maintain slices of the gland, but we can keep them alive. And this is something that we can use for cell culture. And then we can ask questions specifically related to our projects, and we can trial different things such as drugs. And I think later on, we're going to go into the tissue culture room and talk a bit more about that.
Great, thank you very much, Sonia. Okay, so I'm just going to move us from the histology lab now back into the main lab, and then we're going to go and have a look at the microscope room. So once we've prepared our samples, as Sonia's just described, we then use these very high-powered microscopes to view the samples. So these microscopes, such as this one that you can see Sophie on here, are incredibly expensive. So you think several £100,000. So as such, we don't own our own microscopes, and instead we use this specialist core to image any of our salivary gland samples. And the beauty of this is that we have expertise, technical help as well. So this allows us to take not only a magnified view that you can start to see here on this image, but it's also a colour-coded view of the salivary glands. And we've got a video that Ellie's going to share with you now, where my postdoc Chechi is telling you a little bit about how she uses confocal in her lab work.
Hi, I'm Cecilia, a postdoc in Elaine's group working on understanding the mechanism of regeneration of salivary gland. One of the tools we use in the lab is the confocal microscope. The microscope allows us to visualize the structure of the tissue using cell-specific fluorescent probe. In this way, we are able to identify the different type of cells present in the tissue and how their number, position, and interaction with other cell type changes upon irradiation treatment or during regeneration. The confocal microscope, differently from other microscopes, uses a laser system that scans the tissue with high resolution in its 3 dimensions and allows us to obtain 3D images. The 3D images derived from the microscope can then be reconstructed and analyzed to give us a more complete picture of the process taking place during salivary gland regeneration.
Okay, so continuing on with the tour. So I'm just going to take us back to the red lab again, and then this time we're heading off to flow cytometry. So sometimes we want to study a particular cell in the salivary glands without receiving the kind of mixed messages from all of these kind of neighbouring cells. So luckily the centre has a dedicated facility for us to be able to do this, where we can use these complex machines that you can see here to analyse different cell types in a mixed cell population. And this is a technique known as flow cytometry. So like the microscopes, these machines are also incredibly expensive, but this core allows us to be able to do the experiments that we want without having to buy our own machine. So John spends quite a lot of his time in this flow cytometry core, and he's now going to tell us a little bit about how he uses flow cytometry in his PhD project. Over to you, John.
Thanks, Elaine. So as Elaine has alluded to already, I'm a third-year PhD student in her lab. And I look at principally the changes in immune cells after a radiotherapy damage, and specifically what are the changes in the macrophage population. And the reason I'm interested in these cells and this effect is because these are responsible for the clearance of dead tissue and the remodeling of the tissue via the outgrowth of blood vessels and nerves during repair. And this has been shown in a lot of different tissues. So once we've separated the salivary gland into single cells, we can mark these cells with different probes, which will distinguish what these cells are and what kind of signals they're expressing on their surface, which can relate to how they interact with other cells and what their function is. So as Elaine mentioned, these machines are able to detect these probes and to pull apart these different cell populations so that we can see what are the changes in the number, type, and expression of these cells after an injury. And this can help us to better understand what the repair response is and what pathways might be changed after damage and how these differ from healthy tissue. And this will hopefully give us pathways or cells to target for regenerative processes to allow us to restore these cells and therefore the salivary gland back to a healthy state.
Great, thank you very much, John. Okay, so we're now going to leave flow cytometry core and we're going to head up to cell culture. So most of the work that I've described so far has involved looking at the salivary glands as a kind of snapshot in time. However, we sometimes want to study how these cells are behaving while they're alive and while they're growing. And we have a highly specialised tissue culture facility to enable us to do this. So you can see this here in these images. This is tissue culture and where we have the specialist equipment that allows us to culture cells without them getting infected by anything that's circulating around in the air. Ella uses tissue culture quite a lot in her work, and she's going to tell us a little bit about how she uses it in her PhD project.
Thanks, Elaine. So hi, I'm Ella, and I've been working as a PhD student with Elaine for the past 18 months. So the work I do with Elaine is really close to my heart because my grandmother actually passed away from metastatic salivary gland cancer when I was younger. So one of the things I really remember was how radiotherapy kind of affected her and her quality of life and eating and speaking. And it's one of the things that really motivates me every day, kind of whilst working on my project. So if I just tell you a little bit about my project. So it's known that in other parts of the body, such as the gut and mostly the gut we've looked at, radiotherapy can cause inflammation, which can make it really difficult for the damaged tissues to heal and repair, which can lead to some of the side effects that patients might experience. So given that we want to regenerate the salivary gland as part of our projects with Elaine, we want to give the stem cells the best possible chance by providing them with an anti-inflammatory environment so that they can work in the best possible ways. So my project works quite closely alongside John's project to look at how signals, but from the nerves, can actually promote this anti-inflammatory environment and which nerve signals are the best at this. So as Elaine mentioned, as part of my project, I use the tissue culture facility quite a lot, and this allows me to isolate specific cells from the immune system, such as the macrophages that John studies, and then I can add different compounds or drugs to see how they respond. So some drugs might actually irritate the immune system and induce the cells to become quite destructive, which isn't something we want a drug to do if we're going to be giving it to a patient. So instead, we actually look for the drugs that can stimulate the immune cells into more healing behaviors so that they can work with the stem cells in our salivary glands to regenerate the gland and hopefully reduce the side effects following radiotherapy.
Great, thank you very much, Ella. So just to give you an idea of what this kind of looks like in real life, I also recorded a couple of videos while I was working in TC last week that I think Ellie is going to share with us now. My intention originally was to try and get a few more of these sorts of videos of us doing kind of the real science in the lab, but as I'm sure you can appreciate at the moment, we're quite restricted by what we can and can't do in the lab at the moment because of social distancing. So this was the extent of what I managed to capture last week, but I hope you'll enjoy seeing this. So here we are in tissue culture. Today I'm cutting up some bits of gland and I'll be culturing them in this incubator here. And this mirrors what would be happening inside the body. So the incubator is set at 37 degrees, and this means that we can study what happens to cells outside of the body. So this is the inside of an incubator, and this is where we grow our cells, and this is supposed to mimic the inside of a human body. Okay, so we're leaving tissue culture now and we're heading back into the general lab area. So the really nice thing about working in a centre like ours, in a lab like ours, is that because of its open plan nature, it's incredibly collegiate and collaborative. If you want to try a new technique or use a piece of equipment that you don't own, it's highly likely that somebody in another group will be able to teach you or show you how to use their equipment. So you can see there's all these bays with this shared equipment, which is really beneficial for us. It means we're then in the best position to be able to make truly exciting discoveries and to be able to use the funding that we've been awarded by charities and government in the best possible way. As you can imagine, this is how it is in a normal situation. At the moment, it's not quite like this. So normally we have a lot of people in the building. We're a building that have over 200 people in on a usual kind of average day. As you can imagine, this is quite difficult at the moment, So it's quite difficult to navigate if you need to learn a new technique or use a new piece of equipment when we're at quite restricted numbers at the moment. This is something that's been quite apparent to Sonia, as she mentioned she started a project basically right at the beginning of lockdown. So it's a challenge for us, but we're managing, I think, fairly well. We're in when we need to be in, and then we all work from home at the times where we're just doing writing or analysis or office work. So it's working not too bad for our group. Right, so after we've done all of this really important work, we need a place to store our samples. And a lot of our samples are really precious for a number of reasons. It's either that they have come from an experiment that was really long, very time consuming, and had used a lot of special or expensive techniques, or even that they've come from a patient who's very kindly donated some of their salivary gland to further our research. So in order to properly store our samples so that they can be either used again or analysed in the future, we store them at very, very low temperatures. So here in this image, you can see a specialist freezer. And this stores samples at -80 degrees Celsius. So this means we can keep them and still use them many years later if we want to, because this freezer is much lower, much colder than what you would have in your house. So this means that the samples are stored for a longer period, and this means that we can go back to them and look at them again in the future if we really need. So finally, I'm just going to switch down to I think is quite an exciting part of our centre. And this is that ultimately we're aiming to try and develop a strategy to regenerate salivary glands in patients who've had glands that have been inadvertently damaged by radiotherapy, surgery, disease. We anticipate that in the future we'll be able to collect patient cells before they undergo their cancer treatment, for example, and then grow them in the lab before transplanting them back into the patient after the treatment is finished. And this hopefully will then regenerate the salivary glands. However, in order for us to be able to do this, we need to be able to keep and grow the cells in a very, very clean lab. So luckily for us, we actually have a facility like this in the building, and you can see that here in this image that I'm showing you at the moment. So this facility is known as a good manufacturing practice, or GMP facility. And as you can see from the staff inside, they're all suited up, keeping the lab really, really clean and free from any kind of contamination is really, really important if we're then to transplant these cells back into patients and for them to have the beneficial effect that we're hoping they'll have. Okay, so we're heading back into the lobby here. So I really hope you enjoyed your virtual tour around the Centre for Regenerative Medicine this afternoon. And at this point, if anybody's got any questions, myself and my team would be really happy to answer them, whether it's about science or the building, anything at all that you'd like to ask us. If anybody wants to put on their cameras and microphones now, then please feel free.
Sonal has raised their hand, Elaine.
Okay.
Yeah, hi, thank you for this wonderful tour. First of all, a big hello from India. I am curious about two things. One is like, I know Roslyn is shaped like a chromosome, the building. Is there something special about the shape at CRM as well? Because it's quite unique when you see it from outside. And the second thing is, I noticed during the tour that it's very well lit. Like, I think during the daytime you wouldn't need to switch on the lights because the sunlight can enter really well. So are any like energy efficient things followed when this building was built 10 years ago, or something special about it as well?
Yeah, so there's a number of things. When the building was built, they actually wanted it to be very sustainable and be sort of as environmentally sustainable as possible. So one of the reasons that it's so well lit is for exactly that reason, so that we don't have to have unnecessary lighting. We have natural, all the offices are on the outside, so we have natural light coming into the places where people are sort of sitting and reading, using computers, and also that we can have windows open. So again, having ventilation, which obviously is really important if you're sitting working for a long time at a computer. And then we have the labs towards the centre, which then makes more sense because when we have everything sort of within a core, I suppose, in the centre of the building. There are also lots of other sort of particularly environmentally friendly parts of the building that were specifically done when they were built. So the architects put those in mind of having a sort of environmentally friendly building, and things like we have all the lights are on sensors, so they'll go off at a certain point when people are not in the building, so we don't have the building lit kind of all day. So yeah, there's lots of different things like that. In terms of the shape, I don't think somebody can correct me if they know something different, but I don't think it's based on anything sort of scientific. But it's an interesting looking building, and it's really nice to work inside as well. So thank you very much for that question. Thank you.
Thank you so much. Sorry, my mic was mute. Thank you.
No worries. Thank you. We've got a couple of questions in the chat. So Suzanne's asking how long do hospitals keep biopsy samples from patients given future changes in technology to re-examine samples? That's a really great question. So we store all of our samples that we collect actually in the building, in that freezer that you saw there. We can keep them up to 10 years, so that's quite a long time. What we sometimes do is we'll collect the samples and actually not process them, so not do anything to them other than store them in the freezer. Because then at that point, if we ever want to do something further down the line, like a new technology, we should just be able to defrost that sample in the same way that you would defrost your pork chops for dinner, and use that tissue. And it shouldn't be hampered by the fact that we've done anything to it beforehand. If we progress too far down a particular avenue, like fixation, so fixing the sample like Sonia was describing, then it might mean that we can't go backwards. We can't do a technique that would require something to not be fixed. So usually if we've got a very precious sample like that, we might just store it at the -80 as it is, and then we can go back to it at any point and see if there's a new technology or a new test that we want to do. We should still be able to use it. Don't know if any of my team have anything if they want to add, please do.
Yeah, I was just going to say as well that you find that as time goes on, it's easier and easier to take a sample that has been fixed or stored for a long time and do something entirely novel on it. So a sample can be taken, even if it's, you know, fixed dead tissue, and can be analysed by new techniques like single cell sequencing just on their nuclei, or looking at their RNA using kits designed to help with fixed tissue.
Super, thank you, John. So Joanna's asking, in the long term, might it be possible to develop cells taken from a patient after radiotherapy and therefore address the issues that have arisen? Yes, that's another possibility. What we're assuming at the moment is that the cells and the tissue itself, the organ itself, is quite badly damaged by the radiotherapy. And that therefore those cells are sort of less of a potential possibility for kind of regenerating the tissue. So that's why our idea is, if we can do it, try and take the cells before you've got the radiotherapy, keep them, grow them in that very clean lab, and then transplant them back in. And that would hopefully prevent anything sort of serious going wrong with the cells that would then impact kind of the future function. Saying that, though, science and medical research is progressing, you know, every single week. There are new technologies coming out. We're finding new things. You know, several years ago, we had no idea that we could change the sort of maturity of a cell, whereas we now know that we can take, for example, a skin cell, which is sort of mature and differentiated into a skin cell. And as far as we were concerned until recently, it was always a skin cell. People have now found that you can take that skin cell and actually revert it back to a more stem cell-like state, and then make that cell into something else. So make it into muscle, make it into spinal cord. So we're finding things out all the time that we didn't necessarily know before. So I would never want to say that we couldn't take cells after radiotherapy and try and revert them back into something better. I think in the long term, that would obviously be an incredible goal for us, and especially for anybody who's already been through radiotherapy, where banking cells prior to treatment actually isn't an option at all. So Rahima is asking, is there anything added to immune cells or macrophages during flow cytometry? John, do you want to take this, given that you're the expert?
Yeah. So nothing in terms of like drugs that we want to see from a response, but of course the process of, you know, taking apart this delivery gland, digesting it, requires enzymes. They're physically pulled apart, and they're sent through a series of buffers. So there are lots of things which alter perhaps what they're expressing and what they look like, but nothing with the intention of changing them, because we're trying to get an idea of the closest to what's naturally going on during the healthy tissue and after the injury. However, we could then take cells we have isolated and add things to them, and we could grow them in tissue culture.
Well, thank you everybody for joining us on this virtual tour today. If anybody thinks of any questions later, feel free to email. I can either answer myself, or I can put you in touch with the relevant person in my team to be able to answer anything kind of more specific. We're also on Twitter, so please do follow us if you're somebody that uses Twitter. And thank you all very much for attending, and I hope that this was a sort of eye-opening experience into what goes on in medical research, and especially for something to do with head and neck cancer. And I hope you all enjoyed the rest of the meeting.